Why does a frog survive in a place where almost everything else would struggle to live? High on the Baw Baw Plateau in southeastern Australia, the landscape can be cold, wet, and unforgiving. Mist moves through the vegetation. Water saturates the ground. Temperatures can drop sharply, and the habitat suitable for one tiny amphibian occupies only a fraction of the mountain. The frog itself is barely 6 centimetres long. Yet this tiny animal has survived a catastrophe that wiped out roughly 98% of its population. A microscopic fungus attacked its skin, populations collapsed, and the species was pushed to the edge of extinction. Today, fewer than 500 wild Baw Baw frogs are believed to remain. And in 2025, conservationists released more than 3,000 young frogs and 40 adults back onto the plateau. Some of those young frogs were barely larger than grains of rice. They were being returned to the same mountain where their species had almost disappeared. But if the deadly fungus is still present, why release them at all? What allows this tiny frog to survive where everything else dies? This is the story of the Baw Baw frog, Philoria frosti. The first thing that makes the Baw Baw frog extraordinary is not something you can easily see. It is where it lives. Philoria frosti is endemic to the Baw Baw Plateau and surrounding areas of Victoria, Australia. That means there is nowhere else on Earth where this species naturally occurs. Its known distribution is extremely restricted, with suitable habitat concentrated in a small high-altitude landscape. For most animals, losing one valley or one forest would not mean losing the entire species. They could move. They could find another river, another mountain, another forest. The Baw Baw frog has almost no such option. Its world is measured in kilometers. Its survival is tied to a mountain. And that mountain has exactly the conditions the frog needs. The Baw Baw Plateau rises to more than 1,000 meters above sea level, creating a cool and exceptionally wet environment. The frog is associated with wet heathland, woodland, forest, gullies, seepages, and saturated ground. This is not a conventional frog habitat built around a large pond. Much of the frog's life happens among the hidden wet spaces of the forest floor. That is one of the first misconceptions about this species. You might expect a frog to need open water, but the Baw Baw frog has evolved a different solution. Its breeding sites are often small natural cavities associated with vegetation, rocks, and fallen timber. Females lay eggs in foam nests hidden within these wet structures. Depending on the individual and conditions, a clutch can contain dozens of eggs. The eggs develop inside the protected nest, and the larvae can remain associated with this tiny reproductive environment before eventually completing metamorphosis. The mountain itself becomes part of the reproductive system. Moisture is trapped beneath vegetation. Water moves slowly through the soil. Small seepages create constantly damp pockets. The frog does not need a huge pond when it can exploit a miniature world that remains wet for long periods. And that tells us something important about Philoria frosti. It is not simply a frog that happens to live in the mountains. Its entire life cycle is adapted to a very particular environment. Its body, behavior, and reproduction are connected to the cold, wet landscape around it. But that specialization comes with a price. If the mountain changes, the frog has very few alternatives. And then something arrived that did not need to change the mountain at all. The greatest threat to the Baw Baw frog is not a predator with teeth or claws. It is a fungus so small that a human being cannot see it. Batrachochytrium dendrobatidis, usually called BD, causes the disease chytridiomycosis. It has devastated amphibian populations around the world and is one of the most important causes of amphibian decline ever documented. For a frog, the skin is not simply an outer covering. It performs functions essential to life. Amphibians can absorb water through their skin, and their skin plays a major role in maintaining the balance of salts and electrolytes inside the body. BD attacks that system. The fungus infects keratinized skin and can interfere with the normal movement of electrolytes across the skin. In severe infections, this can disrupt the electrical activity of the heart and ultimately kill the animal. The frog cannot simply stop using its skin. It cannot hide from the fungus in a way that removes the biological problem. The organ that makes life possible can become the route through which disease destroys the animal. For the Baw Baw frog, the consequences were devastating. The species suffered a population decline of around 98%. That number is difficult to comprehend. Imagine a population of 100 frogs becoming just 2. Now imagine that happening across an entire species already restricted to a single mountain landscape. This was not a slow decline over centuries. The chytrid outbreak transformed the conservation status of the frog. The species that once occupied much more of its available habitat was suddenly reduced to scattered survivors. And yet something strange happened. The species did not disappear completely. Some frogs survived. Those survivors became incredibly important because they represented something more than individual animals. They represented the remaining genetic foundation of an entire species. But why did they survive when so many others died? There is no confirmed single superpower that makes the Baw Baw frog immune to chytrid fungus. In fact, the disease remains its primary threat. The answer is probably much less dramatic. Some individuals survived because of a combination of biology, location, environmental conditions, and chance. In a species this rare, survival does not have to mean being stronger than the threat. Sometimes it simply means that enough individuals remain alive to reproduce. To understand why those survivors mattered, you have to understand the animal itself. Philoria frosti was formally described in 1901 by the Australian zoologist Walter Baldwin Spencer. The species was named in honor of Charles Frost, a naturalist associated with early collecting and study of Victorian amphibians. It belongs to the family Limnodynastidea and the genus Philoria. The frog is small, compact and generally brown in appearance, with a body adapted to life among wet vegetation and forest debris. It is not an animal that immediately looks extraordinary. There are frogs that are brighter, larger, and more visually dramatic. But appearance tells us very little about survival. The Baw Baw frog's real speciality is its relationship with its environment. It spends much of its life concealed among rocks, vegetation, logs, and saturated ground. Its breeding behavior is particularly unusual. Rather than depending on large open ponds, females create foam nests in protected wet cavities. The eggs hatch inside these structures, and the larvae can develop in conditions that would look extremely restrictive compared with the ponds used by many other frog species. The tadpoles have unusual developmental characteristics, including the absence of typical larval tooth rows. They also retain substantial yolk reserves that help support them during development. Researchers have documented larvae consuming material associated with their eggs and nest environment, including jelly and dead biological material. The young are therefore developing in a highly self-contained system. Everything about the process reflects the environment in which this frog evolved. The reproductive site is small. The water supply is limited but persistent. The nest provides protection. The young can complete a substantial part of their development without needing a large open body of water. It is an elegant solution to life on a mountain. But evolution can create a dangerous trade-off. The more precisely an animal is adapted to one environment, the harder it can be for that animal to survive when conditions change. The Baw Baw frog has no large geographic safety net. There is no distant population in another country. There is no alternate mountain range. If the conditions of the plateau become unsuitable, the frog cannot simply pack up and leave. And the threats were not limited to disease. The Baw Baw Plateau has experienced human influence as well. Historical logging altered parts of the landscape, while development and recreational activity created additional pressure on habitat. Climate change adds another problem because this frog depends on a cool, wet mountain environment. A few degrees of temperature change may sound insignificant to a human standing outside, but for an amphibian whose reproduction depends on specific environmental conditions, temperature can influence development, growth, and survival. Researchers have studied the effects of temperature and natural substrate on Baw Baw frog larvae, including experiments at temperatures of 12 and 17 degrees Celsius. The results help reveal how environmental conditions influence larval development and growth. That kind of research matters because conservationists cannot simply release captive bred frogs and hope for the best. They need to know what the animals require before, during, and after release. The challenge becomes even more difficult when the primary threat is still present. The fungus has not disappeared from the mountain simply because humans began breeding frogs. The disease remains part of the environment. This means conservation cannot be based on the assumption that the threat will eventually go away. Instead, scientists have had to develop another strategy: create enough frogs to give the species a better chance of surviving the threat. That sounds simple. It is not. Captive breeding a rare amphibian is difficult enough. Producing animals that can eventually survive independently in the wild is much harder. A captive frog lives inside a controlled environment. Temperature can be monitored. Food can be provided. Predators are excluded. Disease can be managed. In the wild, none of those conditions are guaranteed. A frog released onto the plateau has to find shelter, find food, avoid predators, survive weather changes, reproduce and deal with pathogens. Conservationists therefore had to learn how to reproduce the Baw Baw frog reliably. They had to understand its breeding cycle, its reproductive behavior, its eggs, its tadpoles, and the conditions needed for successful development. In 2010, a dedicated conservation breeding program was established for the species. The purpose was to maintain an assurance population outside the wild and prevent the species from disappearing completely if the remaining wild population collapsed. The program became a form of biological insurance. But insurance is only useful if the population can actually grow. Breeding the Baw Baw frog required more than placing two adults together. Researchers have investigated hormonal techniques to stimulate reproduction and improve spawning success. Work published in recent years has examined treatments involving gonadotropin-releasing hormone analogues and metoclopramide, with different protocols producing different spawning rates. In one study, 71% of male-female pairs spawned under one treatment, compared with 57% under another and 33% in controls. These numbers may seem small until you understand the scale of the problem. When you are trying to recover a species with a tiny wild population, every successful breeding event matters. A few additional clutches can mean hundreds of future animals. A failed breeding season can mean losing an entire generation of potential recovery. Researchers have also investigated sperm collection and cryopreservation. Preserving sperm allows genetic material to be stored for future breeding efforts and reduces the risk of losing genetic diversity if the living population becomes smaller. This is conservation at a completely different scale. The animal may be only 6 centimetres long, but saving it requires genetics, reproductive biology, veterinary science, ecology, and field monitoring. The goal is not simply to keep frogs alive inside a laboratory. The goal is to produce frogs capable of living on the mountain again. That distinction is crucial. A conservation breeding program can maintain a population for years, but a species is not truly secure until it can survive without permanent human support. And that is where the story reaches its most dramatic moment. After years of captive breeding, the frogs began returning to the place from which their species had almost disappeared. In 2025, conservationists released more than 3,000 Baw Baw froglets and 40 adult frogs onto the Baw Baw Plateau. It was one of the largest release efforts ever undertaken for the species. The scale is remarkable when compared with the remaining wild population. Some of the released froglets were approximately the size of grains of rice. Think about what that means. These are not animals that look capable of surviving on a hostile mountain. They are tiny, vulnerable, and almost impossible to notice. Yet thousands were deliberately placed into the wild. The adults were fitted with small radio transmitters so researchers could track their movements and monitor how they used the habitat. Detection dogs were also used to help locate frogs hidden beneath vegetation and leaf litter. This is not a simple release and forget operation. It is an experiment that may take years to evaluate. The froglets need to survive long enough to reach adulthood. The adults need to establish themselves and reproduce. The released population needs to interact with the environment without collapsing from disease. And eventually, conservationists need evidence of successful reproduction in the wild. The first photograph of thousands of froglets disappearing into the mountain is impressive. But it does not prove recovery. The real test comes later. How many survive their first year? How many reach maturity? How many call? How many breed? How many offspring appear in the next generation? And above all, how many survive in a landscape where BD is still present? That is why the 2025 release should not be seen as the ending of the story. It is the beginning of a new experiment. Humans have spent years protecting and breeding the frog. Now the mountain has to decide what happens next. The frogs must live without constant human intervention. They have to become wild again. And that means facing the same disease that caused the original collapse. Chytrid fungus is a particularly difficult enemy because it does not behave like a traditional predator. You cannot scare it away. You cannot watch it approaching. You cannot build a fence around the frog and expect the threat to stay outside. BD exists at a microscopic scale, and its infectious stages can move through wet environments. For an amphibian living in saturated mountain habitat, avoiding water is not an option. The frog's environment and the pathogen's environment overlap. That is why the disease has been so devastating to amphibians worldwide. In many cases, it can move through populations and cause rapid declines. For the Baw Baw frog, geography made the problem worse. A widespread species might lose one population but retain others hundreds or thousands of kilometers away. Philoria frosti cannot rely on that safety net. Its entire natural range is concentrated in the Baw Baw region. If disease pressure becomes too high across the plateau, there is no untouched second population waiting elsewhere. This is the brutal mathematics of endemism. Every habitat patch matters. Every breeding site matters. Every adult matters. And every froglet matters. But disease is not the only pressure. Climate change could alter the conditions that make the plateau suitable for the species. The Baw Baw frog depends on cool, moist environments, and changes in temperature and rainfall can influence the availability and quality of breeding habitat. Climate change can also interact with other threats rather than acting alone. A landscape altered by development or historical logging may respond differently to changing weather than an intact habitat. A species already weakened by disease may have less capacity to absorb another environmental stress. This is why conservationists cannot solve the Baw Baw frog problem with one action. Captive breeding alone is not enough. Habitat protection alone is not enough. Disease research alone is not enough. The species needs all of them at the same time. And that makes the frog's recovery extraordinarily difficult. But it also reveals something important about how endangered species are actually saved. There is rarely one heroic solution. Recovery is usually a chain of small victories. A breeding pair produces eggs. Researchers learn how to improve survival. A tadpole reaches metamorphosis. A froglet survives. Genetic material is preserved. A habitat is protected. A released adult establishes itself. A male begins calling. A female produces another nest. Another generation appears. None of these events looks dramatic by itself. Together, they can determine whether a species continues to exist. That is especially true for the Baw Baw frog because its reproductive biology is so specialized. The female's foam nest is not just a place where eggs happen to be deposited. It is a carefully adapted solution to the conditions of the mountain. Eggs can develop inside a protected, moist cavity rather than floating in a large pond. Tadpoles can remain associated with that environment while developing. The young eventually metamorphose and emerge into a world of dense vegetation and wet ground. For most of the year, the frogs can be extremely difficult to find. Then, during the breeding season, males call. Recent research describes calling activity from approximately September through March, with a peak around October and November. For scientists, these calls are crucial. A hidden frog is difficult to count. A calling male provides evidence that the animal is alive and potentially participating in reproduction. The sound of a frog can therefore become a population measurement. It tells researchers that the species is not merely surviving in captivity but is still attempting to reproduce in its natural environment. That is the difference between preservation and recovery. A collection of frogs in laboratories can preserve a species temporarily. A population that breeds on its own is something else entirely. It is an ecosystem participant. It is a living part of the mountain. And that is ultimately what conservationists are trying to restore. There is another reason the Baw Baw frog matters. It represents something much larger than one species. Amphibians are among the most threatened groups of vertebrates on Earth. Their permeable skin, dependence on moisture and sensitivity to environmental change make them vulnerable to pollution, habitat destruction, climate change and infectious disease. When an amphibian population collapses, it can be an early warning that an ecosystem is under pressure. The Baw Baw frog is therefore a kind of biological indicator. Its survival depends on the continued existence of a very specific mountain environment. Protecting that environment benefits more than one frog. Wet forests, alpine and subalpine vegetation, seepages and mountain waterways support entire communities of plants and animals. The frog is one small part of that system, but its decline tells us that the system itself has been exposed to serious pressure. This is why the story cannot be reduced to saving a cute animal. It is about whether a highly specialized ecosystem can continue functioning under multiple modern pressures. The frog's future depends on disease management, habitat protection, scientific research, and the ability of the population to reproduce. It also depends on something less tangible: time. Conservation cannot force a species to recover overnight. A froglet released in 2025 may need years before it contributes to the breeding population. Its descendants may be the animals that determine whether the program succeeds. The results may not be visible immediately. That can be frustrating because modern conservation often produces numbers that sound impressive: thousands of frogs released, hundreds bred, genetic material stored. But the ultimate measurement is simple. Are wild frogs still there decades from now? Are they reproducing? Is the population stable? Can the species survive without being constantly rescued? Those questions remain open. And that brings us back to the original mystery. Why does this frog survive where everything else dies? The answer is not that Philoria frosti is invincible. It is not immune to chytrid fungus. It is not protected from climate change. It cannot escape habitat loss. It is not large enough to overpower a predator, and it cannot move hundreds of kilometers to find a safer home. Its survival is much more fragile than that. The Baw Baw frog survives because enough individuals manage to persist through a catastrophic decline, because scientists captured and bred animals before the species disappeared, because researchers learned how to reproduce them more effectively, because genetic material has been preserved, and because thousands of young frogs are now being given another opportunity to live in the wild. In other words, its survival is not a single miracle. It is a chain. Break enough links and the species disappears. Strengthen enough links and recovery becomes possible. That may be the most important lesson hidden inside this tiny frog. Nature does not always reward the strongest animal. Sometimes it rewards the animal that can remain in just the right place, at just the right time, for just long enough to reproduce. The Baw Baw frog found such a place on one Australian mountain. Then disease nearly erased it. Humans intervened. And now the experiment is moving back into the wild. Thousands of tiny froglets have disappeared beneath the vegetation. 40 adults have been released. Radio transmitters are tracking some of them. Researchers are watching, listening, and waiting. But the final answer will not come from the number of frogs released. It will come from what happens after the humans leave. Years from now, the most important evidence may be almost invisible. A small movement beneath a fallen log. A nest hidden inside a wet cavity. A froglet emerging from vegetation. Or, perhaps most importantly, the sound of a male calling on the mountain during the breeding season. That call would mean the chain has continued. A captive bred animal has become a wild animal. A wild animal has survived. A survivor has reproduced. And another generation has begun. The Baw Baw frog is still critically endangered. Its greatest threat, chytrid fungus, remains. Climate change, habitat pressures, and its extremely restricted range continue to make the species vulnerable. Nothing about its future is guaranteed. But extinction has not happened yet. That matters. Because extinction is permanent. Recovery does not have to be. A species can fall by 98% and still return. A frog smaller than your hand can become the center of one of Australia's most difficult conservation programs. And a mountain that once seemed to be the last refuge of a disappearing species can become the place where that species begins again. So perhaps the better question is not why this frog survives where everything else dies. Perhaps the real question is how much a species can lose and still find a way forward. For the Baw Baw frog, the answer is at least 98% of its population. It lost almost everything. But not everything. And that tiny remainder was enough to give an entire species another chance. Now the future of Philoria frosti depends on what those thousands of tiny frogs do next. If they survive, grow, call and reproduce, the mountain may slowly become louder again. If they fail, the Baw Baw frog could move one step closer to disappearing forever. So if you could protect only one part of this frog's future, would you focus on fighting the chytrid fungus, protecting its mountain habitat, or expanding captive breeding programs? Which would you choose? Let me know in the comments. And if you want more stories about the rare animals fighting to survive on the edge of extinction, subscribe.